Hello everyone, a warm welcome to everyone. I am Manish Kumar Dubey. I am a faculty at Galgotia University at the Department of Life Sciences and under the School of Basic and Applied Sciences. And today we are going to discuss about the topic called ribosome, in which we mainly study about the structure and function of ribosome. And this topic will come under the course name called cell biology. So the main objective of our today's lecture is to study the structure and function of ribosomes. Now in the introduction, first of all, we want to know about what is ribosome. So ribosomes are minute cellular non-membranous particles that have a very small diameter, roughly around 23 nanometer. And it was first of all observed by Robinson and Brown in the year 1953 in bean roots, whereas if we see the animal cells, it was mainly observed by Pallade in the year 1955 in the animal cells, and that's why it is also called as Pallade particles. Ribosomes forms a major fraction of microsomes. So what is microsome? Microsome are mainly made up of broken component of endoplasmic reticulum, ribosome and Golgi bodies. And this microsome term, it was given by Clade. Ribosome is also known to us through other name like ribonucleic nucleoprotein. What is ribonucleoprotein? Ribonucleoprotein, it is the name that is derived by the combination of ribosomal RNA and proteins, and in short we are also known as ribosome as RNP particles. Let's talk about the origin of these ribonucleic particles. If we see the eukaryotic cell, they have the different location about there is a different location about the origin of these ribonucleic particles. Like the ribonucleic acid that is present in the RNA, that is mainly gets synthesized in the nucleolus region that is present within the nucleus in eukaryotic cell, and all the major fraction of these ribonucleic acid or ribonucleic particles, they are formed by RNA polymerase first. And what are those portions? Like 28S, 18S and 5.8S, they are all synthesized by RNA polymerase first, whereas there are two other type of RNA polymerase as well that is found in eukaryotic cell, that is RNA polymerase second and third. The another important component that is called as 5S ribosomal RNA, it is synthesized mainly by RNA polymerase third, whereas the RNA polymerase second in these eukaryotic cell, they are mainly deal with the synthesis of messenger RNA, heteronuclear RNA and small nuclear RNA. And the last one, that is the RNA polymerase third, it mainly deal with the synthesis of transverse RNA. And these RNA, they are different from each other on the basis of their sensitivity towards the towards the toxin that is called as amnitin. Like RNA polymerase is have greater sensitivity towards no sensitivity towards amnitin, whereas RNA polymerase second, they have high sensitivity towards the amnitin toxin, whereas the last one, the RNA polymerase third, it has moderate sensitivity towards the amnitin toxin. Now, if we see the prokaryotic structure, then we will find that these RNA ribonucleic particle, they mainly get originated in the cytoplasm because there is no membrane nuclear envelope inside the prokaryotic cell. So they are mainly synthesized in cytoplasm. And here, unlike the eukaryotic cell, we can see that there is a single RNA polymerase that is responsible for the synthesis of all the ribosomal RNA. Not only all the ribosomal RNA, but other RNAs as well, like messenger RNA and transverse RNA. And it is well known that there are around 7,000 RNA polymerase uh, enzymes that may be present in a E. coli cell, and probably there are around 22,000 to 5,000 enzymes that is synthesizing for a RNA at one time. But this number, it can vary depending upon the growth condition of the E. coli. Now, if we see the numbers, So their numbers vary both in prokaryotic and eukaryotic cell. It roughly approximately they have 5 lakh ribosome per cell, and their diameter it range from 152 to 50 angstrom. And if we see the size is much smaller of these ribosome in the prokaryotic cell. It is around 29 into 27 nanometer, and the molecular weight is also smaller in prokaryotic cell. It is around 2.7 into 10 power 6, and the prokaryotic ribosome, it is mainly formed by the major fraction of these prokaryotic RNA. It is formed by ribosomal RNA, and it contain around 60 to 65% ribosomal RNA and 30 to 40% of protein. In contrast, if we see the eukaryotic cell, their ribosomes they have slightly larger, that have a diameter around 34 nanometer, and the molecular weight is also higher as compared to the prokaryotic ribosome. It is around 4 into 10 power 6, and the major fraction of, unlike the prokaryotic ribosome, here the major fraction is formed by the protein, and which is around 55%, whereas the RNA here is around 45%. And they are mainly located, if we see the location where they are present inside the cell, it is around cytoplasm. But if we see the prokaryotes, they are mainly present in cytoplasm in the free state. And if we see the eukaryote, they are present in cytoplasm either bound with endoplasmic reticulum or freely suspended in the cytoplasm. And here in cytoplasm, they mainly bind with endoplasmic reticulum with the help of receptor that is called as ribofourin. To the larger subunit of these ribosome, they get bind with the endoplasmic reticulum. There are certain 70s type of ribosome that is also present in mitochondria and plastids, and these ribosomes they are known as organelle ribosome as well. Now, if we see about the composition of these ribosomes, then we will find that lipid is totally absent in these ribosomes, and they constitute about 70 to 75% of the total RNA content of any cell. And these ribosomes, as we know, they are of two types. One is 70s type, that is predominantly found in prokaryotic organism, and they are also called present in certain organelle of eukaryotes like mitochondria and plastids, and they mainly consist of ribosomal RNA that is around 60 to 64% and contain a much smaller fraction of protein that is around 36 to 37%, whereas the other ribosome it is called as ATS type ribosome, and it is mainly present in eukaryotic organisms. And here the total ribosomal RNA content it is around 30 to 37%, which is very less as compared to the 70s type ribosome. And here the protein content is slightly higher, which is around 56 to 60%. Now, if we see the general structure and type of ribosome, as we know the ribosome are of two types, one is 70s type and other one is 80s type. And these 70s type ribosome, it is made up of two subunit. What is these two subunit? It is a large subunit that is called as 50s and the smaller subunit that is called as 30s. And these 30s subunit it is mainly made up of 16s ribosomal RNA and 21 proteins. When these 16s ribosomal RNA it will combine with 21 protein, it will lead to the formation of 30s or small subunit of 70s ribosome. Whereas the large subunit of these ribosome they are mainly formed by the combination of 28s ribosomal RNA plus 5s ribosomal RNA along with the proteins. And here we already discussed that these 5s ribosomal RNA it is encoded by RNA polymerase third. Similarly, if we see the ATS ribosome that is predominantly a eukaryotic present in eukaryotic cell, it is made up of two type of different component of ribosome. The larger component it is called as 60s and the smaller component it is called as 40S. So the larger component, it is made up of three different ribosomal RNA, that is 28S, 5.8S and 5S, and these different ribosomal RNA, they will combine all together with around 50 protein and form the large subunit, whereas the small segment of these RNA, that is called as 40S 40S subunit, it is mainly formed with 18S ribosomal RNA when it combined with around 33 proteins. 80S ribosome. How the ribosomal protein and ribonucleic acid, they will combine together to lead to the formation of 80S ribosome. So here we can see there are two subunits, that is one is large subunit in the case of 80S ribosome, that is made up of, that is also called as 60S subunit, whereas the small subunit, that is also called as 40S subunit. So this large subunit, it or the 60S subunit, it is mainly made up of 49 ribosomal protein, and when this 49 ribosomal protein, it will combine with 5S ribosomal RNA, 5.8S ribosomal RNA and 28S ribosomal RNA, the combined complex structure that will lead to the formation of 60S subunit. Similarly, when 33 ribosomal protein, that is synthesized inside the cytoplasm, they will combine with 18S ribosomal RNA. It will lead to the formation of a small subunit, that is also called as 40S subunit. And when these 60 and 40 subunit combine together, it will lead to the formation of 80S ribosome. Fine structure of these ribosome. It was mainly described with the help of Lake model, that is recently given by James A. Lake in the year 1981. And according to this model, the different subunit of ribosome, that is small subunit and large subunit, that is made up of three different components each. So if we see the small subunit, it is mainly of three different components, that is called head, base and platform, and the platform is separated from head and base with the help of clef. Similarly, the large subunit, it is also composed of three different areas, that is called ringe, central protuberance and stalk, and the central protuberance region is separated from ringe and stalk with the help of a region called valley, and when these head, it will unite with the central protuberance region of the large subunit, it will lead to the formation of a complete functional ribosome. Ribosome. The ribosome mainly get assembled inside the cytoplasm, and for the assembly of ribosome, the concentration of magnesium ion is very important. If the concentration of magnesium ion is around 0.001 molar, then what will happen? The small subunit and large subunit, they will assemble over each other and lead to the formation of a functional ribosome that is capable of doing the protein synthesis. However, if the concentration get decreased from this 0.001 molar, then what will happen? These two subunits get disjoined and the protein synthesis inside the cell, it will get halt. So ribosomes are also known as protein factories of the cell. That's why because for translation process to occur, the assembly of ribosome, it is very important phenomena. And when the concentration of these magnesium ion, it will get increased tenfold inside the cytoplasm, then what will happen? It will lead to the formation of dimer. Like what will happen? There is a formation of 120S dimer. Two different ribosomes, they will unite with each other and the process will of protein synthesis get halt. So it is also observed that during the process of protein synthesis, 4 to 5 ribosomes, they are present in a group over messenger RNA. And when they are present in a group of 4 to 5, these type of ribosomes, they are also known as polyribosome or polysomes. And they are mainly get attached with these messenger RNA that is synthesized over these or whose sequence is encoded by the over this ribosome. It mainly get attached with the small subunit of uh, ribosome. So if we see inside the ribosome, when 50S subunit and 30S subunit, they will combine with each other, it will lead to a formation of different sites. So one of the most important site is amino acid site. It is denoted by a letter A. The other side is peptidyl site. It is denoted by the letter P. And the last site is exit site. It is denoted by the letter E. And there is a messenger RNA binding site from which the information that is encoded inside the messenger RNA get transformed into a protein. So when in amino acid site, what will happen? There is the transfer RNA will come and it will decode the codon that is present inside the messenger RNA. Similarly, in peptidyl site, there is a formation of peptidyl bond take place, and in the exit site, what will happen? The transfer RNA will get out of the complex, and we will, as more and more transfer RNA will come, there the peptide chain will grow and more protein synthesis will take place. Now, if we see the binding of ribosome to a uh, endoplasmic reticulum, then how the ribosome get decide that they have to remain free in cytoplasm or they have to bind with the endoplasmic reticulum. So what will happen when the cytoplasm get assembled inside a ribosome get assembled inside a cytoplasm, then what will happen? They will start producing a polypeptide, and these polypeptide, they have a special signal, that is when they produce a nascent polypeptide, these polypeptide act as a signaling molecule, and it will interact with a signal recognition protein that is also present inside our cytoplasm. And when these nascent polypeptide, it will combine with the signal recognition protein, it will help the whole complex to bind with the ER membrane. And once the signal recognition protein, it will get bind with the signal recognition receptor, then what will happen? This signal recognition protein, it will get detached, and the process of protein synthesis, it will take inside the ER lumen, means the polypeptide, it will get now internalized inside the ER lumen. Now let's see the difference between the prokaryotic ribosome and eukaryotic ribosome. If we see the prokaryotic ribosome are mainly found freely suspended in a cytoplasm, whereas these eukaryotic ribosome, they are mainly found attached to the nuclear membrane or attached to endoplasmic reticulum, along with being freely suspended. And these prokaryotic ribosome, they are mainly found in prokaryotes, that is mainly bacteria and archaea, whereas these eukaryotic ribosome, they are mainly found in eukaryotic organism like animal, plant, fungi and other unicellular eukaryotes. If we see the molecular mass and diameter of prokaryotic ribosome, it is smaller as compared to the eukaryotic ribosome. Similarly, the sedimentation coefficient of the prokaryotic ribosome, it is around 70s as compared to 80s in the case of eukaryotic ribosome. And the prokaryotic ribosome, it is made up of two different subunits, that is 50s and 30s. In comparison, the eukaryotic ribosome, it is made up of 60s and 40s subunit. However, if we see the last subunit of the prokaryotic ribosome, it is formed by the union of two different ribosomal RNA, that is 23s ribosomal RNA and 5s ribosomal RNA. Whereas the eukaryotic ribosome have mainly made up of three different ribosomal RNA molecule, that is 28s ribosomal RNA, 5.8s ribosomal RNA and 5s ribosomal RNA. The prokaryotic ribosome, they are mainly made up of ribosomal RNA, which constitute about 60% of their total weight, and they have a small content of ribosomal protein, that is around 40%. However, the eukaryotic ribosome, they have major fraction, that is mainly formed by the ribosomal protein, that is around 60%, and a small fraction around 40%. Now in the next slide, we will see now the function of ribosome. So ribosome have different function. One is, we have already discussed the ribosome according to their location. They are of two type. One is bound ribosome and another one is free ribosome. So bound ribosome, they are either bound with endoplasmic reticulum or nuclear membrane. So what is the function of these bound ribosome? They mainly secrete the protein, they mainly produce the integral membrane proteins, they mainly produce soluble protein that reside within the compartments of endomembrane system like ER, Golgi complex, lysosome, endosome, vesicle, and plant vacuole. So the protein that are present inside all these endomembrane system, they are synthesized mainly inside the bound ribosome that are either bound to endoplasmic reticulum or the nuclear membrane. The other one is free ribosome, that is mainly remain freely suspended inside the cytoplasm, and they mainly produce the protein that is designated to different portion of the cytoplasm. Mainly it function in the cytoplasm as a enzyme that is responsible for glycolysis, or protein that is useful for the construction of cytoskeleton. They also help in production of different protein that is responsible for peripheral protein, or that constitute the peripheral protein of cytoplasmic surface of the plasma membrane. Similarly, there are many proteins that are formed on this free ribosome, which are get transported to nucleus for different function like expression of genetic information. And lastly and the least is the protein that is incorporated in peroxisome, chloroplast, and mitochondria. They are also produced inside the free ribosome. Thank you everyone.